Hi everyone, Ethan here. Today I'm going to talk about monoclonal antibodies. When I heard about this in lecture, I was immediately like, ooh, that's cool. And apart from the concept itself, I thought that was cool because I have a simple and somewhat misguided belief that big words are good. And I think this really all stems from my time in debate. Because while debate is very complicated, it's also very simple. You only really need to do three things. One, know stuff. Two, make well-reasoned, clear, and effective arguments about the stuff. And three, sound like you know the stuff and are right about the stuff. And because debaters, a lot of them, know more than me and are better at making arguments than me, my specialty, my forte, was just in sounding like I knew what I was talking about. And one of the chief ways of sounding like you know what you're talking about is very simple. It's to use big words correctly. So I hear monoclonal antibodies. Ooh, that sounds interesting. That sounds smart. It sounds something I should learn about. But deeper than this ridiculous justification is that it sort of rung a bell, right? And it's true. During the pandemic, monoclonal antibodies were a therapeutic option used to treat patients with COVID. And that sort of was a familiarity, ring a bell in my mind. So that's sort of the why I chose this topic. Let's talk about the what. What am I going to talk about today? A couple things. What is an antibody briefly? Structure and function of monoclonal antibodies, brief overview and their uses, and an extremely brief overview of the production of a monoclonal antibody. Let's start by talking about antibodies themselves. What are they? They're very versatile ways and specific ways of targeting foreign material. So right here on the left, we see the structure of an antibody. They're large proteins used by the immune system to identify and neutralize foreign substances, such as pathogens. They have two heavy chains seen here, and then two light chains seen on the outer part, and it forms sort of this Y shape, and sort of the tip of this light chain binds specific epitopes, just specific parts of an antigen. An antigen is a specific protein or something that the body recognizes as foreign. And I sort of, again, it's important for me because I was trying to think about this and it was a little confusing to me. If you have a bacteria, right? Imagine you have a bacteria, it might have many different proteins on the cell surface that the body recognizes as foreign. Each one of those proteins is an antigen, and the specific part of the protein that the antibody binds to is the epitope. Individual antigens can have many different epitopes, and actual bacteria or infectious agents can have many different antigens. So again, infectious agent can have many antigens, and each antigen can have many epitopes. An epitope is specifically the specific site on an antigen where an antibody binds to. And that binding part of an antibody, the antigen binding site, where it actually latches onto the epitope, is very variable between antibody and antibody. And that makes a lot of sense if you think about it, right? Because there's a lot of things that we want antibodies to bind to, because there's a lot of infectious threats, a lot of bad stuff coming in from the environment. So we want our antibodies to be able to bind a lot of different things. In order to bind a lot of different things, you better have a lot of different variability on that binding site. When an antibody binds to an antigen, it does a lot of things. Binding of the antibody itself can neutralize an infectious agent, or as seen in this picture here, it can sort of bind to the antibody itself after binding to an infectious agent, can bind to a phagocyte and then initiate phagocytosis. It can activate the complement system. It can do a lot of stuff. And in terms of how these antibodies are created, well, they're created by plasma cells, which are a type of B cell. So when an epitope from an antigen binds to a B cell receptor, seen here, it causes the B cell to differentiate into a plasma cell, seen here. That plasma cell cranks a lot of antibodies that target the specific epitope of the antigen that bound to that receptor initially. And again, I want to really reemphasize, this is a vast oversimplification of antibodies and immune response. I haven't touched antibody classes, domains, interplay with MHC, I haven't touched a lot of stuff, the most of the stuff. I just want to provide some context before I talk about monoclonal antibodies. But for some additional context, I want to talk first about polyclonal antibodies, which comprise the main antibody response in an infection. So recall what I just told you, right? How are these antibodies created? Well, the epitope binds B cell receptor, causing the B cell to turn into plasma cell to create specific antibodies, right, to that epitope that bound. But recall that these foreign elements that we're worried about, they're not like an epitope floating in space. Remember, I told you, again, there's many epitopes, a single antigen, many antigens on a single bacteria, right? So when we look at an infectious agent, let's say SARS-CoV-2, there's a lot of different antigenic targets and a lot of different epitopes on each one of those antigenic targets. So what does that mean? All these epitopes are going to bind different B cell receptors, meaning that those B cells will turn into different plasma cells and crank out different antibodies. And while those antibodies may all be targeting the same broad target, the virus, they're all going to look different and attach the virus on different spots, different epitopes, right? And recall, each antigen can have many different epitopes too. So it doesn't have to just be the virus itself. It can be just an individual antigen. So in this way, the antibody response to a single foreign element is polyclonal. It involves multiple groups of different antibodies. They all bind the same broad target, but at different spots. Monoclonal antibodies, as the name suggests, are different. They are all identical, originating from a single plasmid lineage, and they're made to target a specific epitope. Slight note, there are newer bispecific monoclonal antibodies that can target 2 epitopes, but the core idea is the same. They're all exact copies. It just turns out that their antigen binding site, seen here, happens to be able to bind to 2 epitopes, just like a lock that can be opened by 2 keys. Monoclonal antibodies, their hyperspecific targeting seems less effective though, right? And this is just me, perhaps. Again, I'm not an expert. This is just how I think about it in my mind. Let's say I get infected and I'm trying to neutralize this infectious agent. I want to bind all over the infectious agent, not at just one epitope, right? Because you want to hedge your bets. Why pick one horse in a race when you can pick 10? And you'd be right. This logic or I should say, I would be right. This logic is sort of underlies what we saw during the pandemic, in that viral evolution, the changing of a virus, causes certain monoclonal antibody drugs to be effective, assuming because the evolution of this virus causes a changing of epitopes. If the epitope's different, the monoclonal antibody has nothing to bind to. But the reason why we care about monoclonals more when it comes to therapy and not polyclonal is because polyclonal antibody responses are inherently unpredictable. They depend a lot on host characteristics. Let's just think about it, right? Each organism will produce a different mix of antibodies to an antigen, right? Depends on your own immune response, your own set of memory cells and the like, right? And while that means that, sure, having different antibodies means more binding to that infectious agent, you're not able to sort of homogenize that polyclonal mixture. And even if you originate from the same organism, the fact that you have multiple antibodies means you have increased cross-reactivity, because more antibodies means more possibility that these antibodies will bind stuff that you don't want them to. Antibodies may have a very specific antigen binding site, right? But that site may fit multiple targets. And if you add in multiple antibodies, you increase the chance that you bind stuff that you don't want to bind and affect the immune response in ways that you don't want to affect the immune response, right? So that sort of underlies the advantage of monoclonal antibodies and segues nicely into the uses of monoclonal antibodies. They provide predictable, specific targeting. Because they're all identical clones, they only target a specific epitope, and therefore you can have trust there won't be variability issues, because in a polyclonal mixture, you have multiple antibodies, you only have one copy in the monoclonal mix, and you don't really worry as much about cross-reactivity because you're only dealing with one antibody. You can really control sort of what that antibody is going to bind to. You're not worried about having like 10 different types or whatever, right? These qualities have made monoclonal antibodies a very promising new class of drugs to treat a variety of conditions. We talked about COVID, right? So again, it makes sense. You can pump out antibodies that target a specific infectious agent that can maybe act if your immune system can't, or before, or to augment your immune response, right? Your own natural production of antibodies. But also it's a cancer treatment too, because think about it. If antibodies are just a way to stimulate the immune response, what if we could use the immune response to attack cancer cells? That's the basic idea of how monoclonal antibodies are used to target cancer cells. You create monoclonal antibodies that target cancer cell-specific antigens on the cancer cells, thereby stimulating an immune response against those cancer cells. And you can see that sort of seen right here. Or you can use it to target stuff like VEGF, meaning you can target growth factors such as vascular endothelial growth factor, which is used by cancer cells to stimulate angiogenesis. So by binding the VEGF, you activate VEGF, inactivate angiogenesis, sort of limit this cancer cell's ability to get new blood vessels and grow further. You also target the antibodies against specific cytokines or other proteins in order to affect the inflammatory process itself. That's the basis for drugs like Humira, which treat autoimmune disease. And I want to be very clear, these are pretty complicated drugs with very complicated mechanisms of action. I'm sure any competent pharmacologist or expert is going to say, Ethan, that's a very bad explanation of these drugs, but that's sort of that in a nutshell, the basic level. You're harnessing the body's own immune response by creating antibodies targeted to the stuff that you want to destroy or interfere with. Lots of potential applications in that way, right? So cancer, infectious, autoimmune, but also its applications in the lab via immunohistochemistry. So if you want to identify a specific protein target, you can create monoclonal antibodies that will bind that target. And you can radio label or fluorescent label those antibodies so that once you release those antibodies around the thing you're concerned about, you can look for the fluorescence or the radioactivity to find out where your target is in a specific thing. So you can see here, ooh, that's where your thing is. It's fluorescing. So just sort of a brief bit on production. How are these produced? Well, they're produced by first exposing an animal to a specific antigen. That creates, of course, a response that creates plasma cells that will create antibodies against that antigen. And then you take those plasma cells and you hybridize them with myeloma cells to create hybridomas. Why do you use myeloma cells? Well, these myeloma cells are essentially like cancer cells, right? They happen to be immortal. And plasma cells, on the other hand, actually do die off after a certain period of time. So by creating these hybridomas that are part plasma cell, part myeloma cell, you actually create immortal hybrids, which are good because if you're going through all this work to try to isolate monoclonal antibodies, you don't want the progenitor cell, the plasma cell, to just die off. So from these hybridoma cells, you got to make sure you select for the ones that were successfully hybridized because the hybridization doesn't always successfully occur. You might have a hybridized plasma to plasma cell, right? So you select for the positive hybrids, the correctly hybridized hybrids, and then you harvest the monoclonal antibodies from those hybrids. And you can keep harvesting monoclonal antibodies from the hybrids as long as those hybrids are preserved because they can grow indefinitely, again, because they're part myeloma cells, which happen to be immortal. So just as a review of what we talked about today, antibodies are proteins that bind epitopes on antigens. They're created by plasma cells, and they stimulate the immune response and can neutralize infectious agents on their own. Polyclonal antibodies are a mix of antibodies binding different epitopes on the same antigen. Also, perhaps on the same infectious target, too. And it's a variable mix. Monoclonal antibodies are identical antibodies created to bind the same epitope, and the use of these monoclonals is to bind specific targets, stimulating the immune response against that target or to regulate the inflammatory response itself. Also, immunohistochemistry, sort of radiolabel or fluorescence labeling specific protein targets. And lastly, production. You create these by fusing specific plasma cells with mortal myeloma cells to create hybridoma cells that produce monoclonal antibodies that you can then harvest. So hope this was sort of informative, sort of useful to you all. These are the sources I sort of consulted. I wish you all the best. Take care. Have a good one. Bye-bye.